Thank you for having me today for this webinar. I'm Jung-Eun Yoon from Seoul National University College of Medicine. Today I'm going to talk about metabolic circuit to support tumor progression. So this is about how cancer manipulates adjacent adipose tissue to exploit fatty acids. So according to Korean Society for the Study of Obesity, cancer risk shows a positive correlation with obesity stage in 2024. And among the cancers associated with overweight and obesity, especially breast cancer shows a unique tumor microenvironment, which is a direct interaction between cancer and adipose tissue. Before we begin, I think we need to share about our previous study. Our research team previously reported that fatty acids derived from adipose tissue can upregulate HIF-1 alpha, which is a key transcriptional factor for proliferation and migration related genes in cancer. So the fatty acids from adipose tissue can promote cancer progression. The first to confirm the previous study, I made two mouse groups, which are CHO dieted group and high-fat dieted group. At 5th week, I injected 41 breast cancer cells, and in 3 weeks of time, I collected tumor and tumor adjacent adipose tissue together. And as a control, I collected non-tumor adjacent adipose tissue, so like this. When I compared them, it was quite obvious that tumor progression was promoted by high-fat diet. And at the same time, the tumor adjacent adipose tissue was shrinking for some reason when it is neighboring to tumor. So it made us hypothesize that tumor can exploit fatty acids from adjacent adipose tissue. Meanwhile, the tumor collected from high-fat dieted group showed upregulated HIF-1 alpha expression and Ki67, which is a cancer proliferation marker expression. And also, the genes related proliferation and migrations, the genes mRNA levels were upregulated in high-fat dieted groups tumor. And when we checked about breast cancer patients, in obese group, the same proliferation and migration related genes were upregulated. And also, the survival rate of obese breast cancer patients was lower than that of normal weight breast cancer patient. So for further study, I thought maybe cancer might secrete some signal molecules like as messengers or adipose tissue. So maybe the signal molecules can manipulate adipose tissue so that more fatty acids can be released from adipose tissue. And for this study, I had to co-culture two different cells, which are cancer cells and ADSCs. ADSC is adipocyte-derived stem cell, which is isolated from adipose tissue. And for this, I used 3D co-culture system. I used PDMS 3D culture chip which is made of PDMS. It's a oxygen permeable material, and when the cells were seeded on the plate at the same time, in 24 hours, the cells aggregate and forms spheroids like that. And in this way, we could remodel the unique tumor microenvironment. So first, I monocultured cancer cells and co-cultured cancer cells and ADSC on the 3D culture chip. And I collected the conditioned media, and then I treated the conditioned media to cancer cells. And just like the previous study, I could find that the lipid derived from ADSC increased HIF-1 alpha expression. And when I removed the lipid from conditioned media by charcoal stripping, the upregulated HIF-1 alpha level was restored. And again, when the lipid mixture was added into the conditioned media, the HIF-1 alpha expression level was upregulated again. So it was quite clear that lipid can upregulate HIF-1 alpha in cancer cells. So now to find what kind of signal molecule is secreted from cancer cells, I compared the two conditional media. And I chose the cytokines increased in co-cultured conditioned media. And also at the same time, I chose cytokines under control of HIF-1 alpha, because previously we checked that lipid derived from ADSC could upregulate HIF-1 alpha. So I chose the cytokines which are regulated by HIF-1 alpha. And here I could find that CCL2 was increased in co-cultured conditioned media commonly. And now to confirm that whether the secreted CCL2 is derived from cancer cells rather than ADSC, I tagged GFP to cancer cells first, and then I co-cultured the cancer cells with ADSC. By FACS sorting, I isolated the co-cultured cancer cells only, and then when I checked the co-cultured cancer cells, the CCL2 mRNA level was upregulated. So it showed that the increased CCL2 secretion was originated from cancer cells, not ADSC. So next, to confirm that whether CCL2 is directly regulated by HIF-1 alpha, first I silenced HIF-1 alpha in the cancer cells and treated lipid mixture. And then the CCL2 mRNA level, which was upregulated by lipid, was restored when HIF-1 alpha was silenced in cancer cells. So it was quite clear that HIF-1 alpha can directly regulate CCL2. And it was confirmed as a protein level by fluorescence immunofluorescence assay. So far, I could find that fatty acids from adipose tissue can upregulate HIF-1 alpha, and then this increased CCL2 secretion from cancer cells. And now I wondered whether CCL2 can change the lipid metabolism of adipose tissue. So I just directly treated neutralizing antibody CCL2 to the co-cultured one. And I collected the conditioned media and checked the fatty acid concentration in the conditioned media. And the upregulated fatty acid level was recovered when CCL2 neutralizing antibody was treated. And more specifically, when I silenced CCL2 or HIF1 alpha in cancer cells, and then when I co-cultured the cancer cells with ADSC, and the conditioned media collected from those co-culture showed recovered fatty acid concentration as well. So it was quite clear that CCL2 derived from cancer cells can change lipid metabolism of ADSC. And now to find how CCL2 changed the lipid metabolism in ADSC, I checked big data here. And in peritumoral adipose tissue of obese breast cancer patients, cytokine receptor interaction pathway was upregulated. And among the genes which are belonging in the pathway, there was a CCR2, which is a receptor of CCL2. So next, I screened representative lipid metabolism related transcriptional factors. And then I could find that our PPAR family shows a positive correlation with CCR2 expression in the peritumoral adipose tissue of obese breast cancer patients. And among the PPAR families, only PPAR alpha showed dramatic increase on the CCL2 treatment. And also PPRE, which is a PPAR alpha promoter reason, its activity was upregulated when CCL2 was treated. So now I checked the lipid accumulation in ADSC. When I treated CCL2 to ADSC, as you can see, the accumulated lipid was decreased, showing increased lipolysis of ADSCs. And this was restored when neutralizing antibody, or GW6471, which is a PPAR alpha inhibitor, was treated. And also the 3-lipase mRNA level was increased by CCL2 treatment and recovered by neutralizing antibody or PPAR alpha inhibitor. And when we checked fatty acid concentration in the conditioned media, as well, the upregulated fatty acid by CCL2 was recovered by neutralizing antibody and PPAR alpha inhibitor. So far we could find that fatty acids can upregulate HIF1 alpha, and then it derive increased secretion of CCL2, and then it upregulates PPAR alpha and lipolysis of adipose tissue. This is a mechanism study. It shows the reason why PPAR alpha PPAR alpha is increased by CCL2. And shortly, PPAR alpha mRNA level was not changed by CCL2 treatment. But when I treated cyclohexamide, I could find out that CCL2 can upregulate the PPAR alpha protein stability, so that increase the PPAR alpha's half-life. So here you can see that CCL2 decrease the ubiquitination of PPAR alpha. And before that, CCL2 can activate phosphor-ERK pathway, and this activated phosphor-ERK pathway can phosphorylate PPAR alpha. And finally, I could figure out that CCL2 inhibit the binding of PPAR alpha and HUWE1, which is a E3 ligase of PPAR alpha. So the final mechanism is like this. When CCL2 is treated, phosphor-ERK pathway is activated, and by the activated phosphor-ERK pathway, PPAR alpha is phosphorylated as well. And then it cannot bind with E3 ligase anymore, so that PPAR alpha stability is increased. So now I wondered whether the cancer progression is suppressed when CCR2 or PPAR alpha axis is inhibited in ADSC. So I just silence CCR2 and PPAR alpha in ADSC, and then I co-culture them. And the result was like this. When cancer cells and ADSC were co-cultured, as you can see, the spheroid roundness was decreased, showing increased migration. But when CCR2 or PPAR alpha is silenced in ADSC, the upregulated migration was recovered. And when I checked the conditioned media from the 3D culture, I could see that the increased fatty acid in co-cultured media is recovered in conditioned media collected from co-culture of cancer and CCR2 or PPAR alpha inhibited ADSC. And when I treated the conditioned media to Transwell assay, the migration result was like this. So when the CCR2 and PPAR alpha axis was inhibited in ADSC, the conditioned media obtained from the co-culture recovered the upregulated migration. So the same, when I treated the same conditioned media to only cancer cells on the 3D culture chips, the result corresponds with the previous results of Transwell assay. And then I collected the spheroids from each 3D culture chips, and I embedded the spheroids into the OCT compound like that. And I sliced them and put them on a slide, and I checked the section of the spheroid. And here, as you can see, the proliferation-related marker, which was increased by a co-culture conditioned media, was recovered by the conditioned media obtained from co-culture of cancer and CCR2 or PPAR alpha inhibited ADSC. So finally, this time, this in vivo experiment I injected 41 breast cancer cells first, and then I injected neutralizing antibody CCL2 for 2 weeks. The result was like this. In high-fat dieted group, again, the cancer progression was promoted, but when I treated CCL2 neutralizing antibody, the tumor progression was suppressed. And also, the collected adipose tissue showed that it is not shrinking anymore next to tumor when neutralizing antibody is treated. So next, I checked PPAR alpha protein level in adipose tissue. And as you can see, in high-fat dieted group, the neutralizing antibody treatment recovered PPAR alpha protein level. And also the 3-lipase mRNA level as well, they were recovered when neutralizing antibody was treated. And this time in the tumor from high-fat dieted group, when the neutralizing antibody was injected, HIF-1 alpha and CCL2 and proliferation marker Ki67's expression was recovered. And it was the same when I checked CCL2 mRNA level. And when I checked proliferation and migration-related genes in the tumor, again, by neutralizing antibody, the gene's mRNA level was restored. So this is a human data. When I checked breast cancer from obese breast cancer patients, I could see that HIF-1 alpha and CCL2 and proliferation-related marker is upregulated. And also in adipose tissue of obese breast cancer patients, the peritumoral adipose tissue showed increased expression of CCR2 and PPAR alpha and lipase ATGL when it is compared to non-peritumoral adipose tissue. So this is a graphical abstract. As you can see, fatty acid from adipose tissue can upregulate HIF-1 alpha, and then it increases CCL2 secretion from cancer. And then PPAR alpha is upregulated in adipose tissue, increasing lipolysis of adipose tissue. So again, fatty acid release can be increased by CCL2. So it makes a vicious cycle in obese breast cancer patients. So she's my advisor, Professor Yang Suk-Chun, and they are my co-authors of this study. Professor Kyung-Hong and Professor Junji Fukuda and Professor Ji Eun-seok and Dr. Do-won Chung and Ms. Ye Sun-son. I'd like to thank all the co-authors and especially my advisor, Yang Suk-chun. This study was published in Advanced Science, so you can get more detailed data from the paper. Thank you for listening.